الله الرحمن الرحيم. الحقيقة أنا بشكر الأستاذة الدكتورة ناجية على دعوتي النهاردة، وبشكرها على المؤتمر المميز. دايما يعني مؤتمرات الأعصاب كتيرة جدا جدا، لكن مؤتمرها هي يعني مميز جدا جدا، وبنستفيد كلنا منه، ودايما بيجيب لنا الجديد في النيوروماسكولار. وبشكرها على المحاضرات الشيقة يعني اللي هي بتقدمها لنا والمشتركين في المؤتمر بيقدموها. دلوقتي now we introduce Dr. Eduardo Malfatti, Professor of Histopathology, Paris University. She talks about stem cell in the research of Duchenne muscular dystrophy. قبل كده وعلى فكرة لو شفناه قبل كده. Welcome, Professor Eduardo.
Hello.
Hello. Welcome. We now introduce Dr. Eduardo Malfatti, Professor of Neurology and Histopathology, Paris University. He talked about stem cell in the research of Duchenne muscular dystrophy.
Thank you very much. I will share my presentation. Can you see it?
Yes.
Thank you very much. So I will make a simple thing. Okay. So thank you very much again for this kind invitation. It's a pity that I couldn't be there with you in presence, but I made it to do it online. So it's a very great pleasure to be with you today. Thank you very much, Nagia, for this invitation. Today I will talk to you about stem cell in the research of Duchenne muscular dystrophy, and I would like firstly to show our location. We are in Paris region, University of Paris Est, and this is our hospital, the Henri Mondor University Hospital, where we have a neuromuscular center, neuromuscular reference center, where we perform muscle biopsy, we perform genetics for muscular disorder, and we have a big research facilities, not only for humans and derived tissue, but also for animal tissue with the veterinary school. And then we have a huge as the EFS, where we do conserve the cells from the patient in order to bioengineer new therapies. In our research group, we really focus on translational medicine, on stem cells, and particularly muscle stem cells that I will detail during my presentation. And I run a group that is called Neuromuscular Clinical and Translational Research. The outline of my presentation will be to remind you the importance and the different function of satellite cell. I will discuss a little bit about Duchenne muscular dystrophy and how satellite cell have been previously used in order to design some therapeutic approach. And then I will describe our work based on this DMT rat with deletion 52 that present an alteration in satellite cell, and the study that we performed in muscle biopsy in order to go into conclusion on how we can extrapolate these results to develop and design new therapy. So as you know, skeletal muscle is a huge organ because it represents 40 to 50% of the body mass. It contains the contractile cells with the myofibrils, and it has the capacity to regenerate. The capacity to regenerate is due to the presence of these cells that are peripheral and that are called the satellite cells. These are stem cells that can be visualized through different markers. This is a PAX7 marker, and that are associated with the membrane of the muscle fibers, and they rely on the plasma layer. With the muscle biopsy, we can put in culture this satellite cell, make them proliferate, and what we see when we analyze a section of AHN stained, that we have some muscle fibers with inside different nuclei, and at the periphery there are probably some satellite cells. These are particularly important because during a process of degeneration, the muscle satellite cell can proliferate and regenerate the fibers. Here you have a regenerating fibers that is to present a big nucleus that synthesize all the contractile apparatus. And what we have by electron microscopy, that we can differentiate and identify this satellite cell that you can see are present under the plasma lemma and are separated by the cytoplasm of the muscle fiber. Why these cells are so important? These cells are so important because one, the muscle degenerates or grow, they can activate and proliferate in order to substitute or regenerate the muscle fibers or to contribute to its growth. Here you have their localization. You have an example of fiber necrosis. The inflammation arrive and eat all the cell, and what we have that the satellite cell that are here proliferate, fuse, and regenerate the muscle fiber. The muscle regeneration have been studied in different models, in humans, in animal models, and we can now see the activation of satellite cell with immunohistochemical marker. Here there are some examples of markers of the satellite cell that can be seen with different techniques in the skeletal muscle. What is very important that now we can intervene on the dynamic of the satellite cell to see their expression of different markers that are marker of differentiation, of activation, and with myogenin and MRF4, we have a marker of differentiation that lead to mature muscle fibers. In Duchenne muscular dystrophy, we assist at the degeneration of the skeletal muscle fiber that is driven by the absence of dystrophin due to mutation in the gene of dystrophin on X gene. This patient not only develop a skeletal muscle disorder, they develop a cardiomyopathy and also muscle, for example, of the gastrointestinal tract are affected. And what we have in this patient, we need help for the respiration at around 15 years. We have a severe cardiomyopathy, and now, thanks to the new therapy and the steroids, what we have that the age of death has been delayed. What we have in the muscle fiber of the patient, the dystrophin that usually make this link, this chain between the protein of the membrane and the F-actin is absent, and we have a myofiber fragility with necrosis that provoke cycle of degeneration and regeneration that are exhausted in the patient. When the regeneration is not more effective, what we have deposition of fibrotic tissue and adipocyte infiltration, and the muscle cannot contract anymore. The satellite cell that you can see also here schematized have been studied in models of Duchenne muscular dystrophy, and in the muscle of this patient what we have that there is some kind of regeneration, but this become exhausted and less effective, and you have a massive fibrosis accumulation with fatty substitution. The fatty substitution is done by this other cell that is the fibroadipogenic progenitor, and now we think, and I will show you with our experiment, that there is an Irker talk with a problem of the satellite cell and a dysregulation of the fibroadipogenic progenitor. And now we have a different approach with novel therapy arriving to the market. One example between other is the gene therapy. With 4 gene therapy, one has been approved in the US by the FDA, but still this regenerative process is not completely understood and cannot completely be reversed. The why myoblast transfer has been proposed before as a possible cell therapy. Thanks to the muscle biopsy, in previous clinical trial, the muscle was produced, it was homogenized, and the cells were put in culture till the production of a complete myotube. After that, pioneer studies in the 90s demonstrated that this fiber can be proteased, they can be injected in the muscle of the patient, and this fiber led to low dystrophin expression. Here you have the figure of the paper published by Shook et al. That did this injection in the muscle of the patient. As you can see here, there was an expression of the dystrophin, but this, although promising, it was not continued. In our lab, we wanted to better focus on the mechanism of regeneration, And to do so, thanks to the work of Valentina Taglietti, we created a new rat model that is ablated of the exon 52. This provokes an out-of-frame mutation with the absence of dystrophin. These mice present a progression of disease that is very similar to the humans, in contrast to other animal models, like for example the MDX mice. And so we decided to study the disease trajectory on the muscle derived from this rat. And what we see compared to the CPK levels in the patient and the rats, that we have a very similar presentation with the 6 months that correspond to the age of 10 years when we have a drop on CPK level. But one reasoning was that some muscle in Duchenne muscular dystrophy are spared, and this is specific for the extraocular muscle. When you examine your patient, you see they are weakening of the muscle, but the extraocular muscle are conserved. And so we wanted to see in the animal model what happened to the extraocular muscle. Here you have a tibialis anterior muscle and the extraocular muscle of the rat, and you can see that in the MD you have a prominent destruction of the tissue and a well-conserved muscle in the DMD patient. And this is demonstrating that these muscles are spared. But why this happened? This happened because what we have in tibialis anterior is that in Duchenne we have a progressive degeneration that is due to the loss of regeneration, while these do not happen in the extraocular muscle, where we have a conserved regeneration in comparison with the other muscles. The extraocular muscle maintain their regeneration capacity, and this is demonstrated by the presence of this satellite cell that express markers of activation that are the Ki67. Here you have the tibialis anterior that degenerate, and here in the extraocular muscle, as you can see, the regenerative capacity is well conserved. We also show that this muscle presents a specific differentiation effect with the extraocular muscle that are conserved, and by single cell RNA sequencing, that is an experiment where we took tibialis anterior muscle of the rat and extraocular muscle. We digested them and we performed then it's the genomics technique. We found a different pattern of expression between the TA that degenerate and the EOM that continue to regenerate in Duchenne. What we found that the different markers were altered and overexpressed in the TA muscle, suggesting that there was a problem with two proteins, P16 and P62, that are two markers that contribute to the senescence of the satellite cell. When we perform the experiment, what we see that the tibialis anterior muscle show a high proportion of senescence marker, and what we have is that the dynamic is completely altered because in association with the muscle regeneration, we have an increase of the muscle stem cell senescence. But what happened in the extraocular muscle? We see that in contrast to the limb muscle, we have a sparing from the cellular senescence, and this is particularly evident thanks to the fact that we demonstrated that there is a high expression of the receptor of TSH. The TSH receptor is highly expressed in extraocular muscle. What is the TSH receptor signaling? The TSH is an hormone produced by the pituitary gland, and it goes to a specific receptor of the thyroid, and when its ligands go to the receptor, the TSH, tell to the thyroid to produce the thyroid hormone that are T3 and T4. This pathway works through the adenylate cyclase thanks to the activation of CAMP that activate the PKA. The extracellular expression of TSHR is something that is known in the cardiac muscle, in the immunity cells, and in the bone, but also in the cancer cell. So what we wanted to do was if we could block the TSA receptor or activate the TSA receptor to see if we can act on the satellite cell to ameliorate the Duchenne muscular dystrophy and render the skeletal muscle of the limbs similar to the extraocular muscle. What we have done, we used two compounds, ML224, that is an inhibitor, and the forskolin, that is an activator. When we set the cells and we treated this with these two compounds with the inhibitor, we see that the myoblasts of the EOM do not proliferate, while when we add the forskolin, this proliferated very well. We also check if there was both response to proliferative and differential effect in the MD muscle, checking for the CMP PKA signaling, and this was the case. As you can see in the wild type and the DMD muscle with the forskolin, we had the same phenotype. So we hypothesized that this stimulator of the TSH, the forskolin, could be a therapy for our rats. That's why we decided to treat the wild type with the empty vector and the DMD with the forskolin, and we observed the mice, the rats solving at different time points with different grid and functional tests. What we found that when we added the forskolin compared to the DMD, we had a partial rescue of the phenotype that you can see also here. And when we examined the skeletal muscle, we found that the DMD plus forskolin reverted their deficit in regeneration and improved the interstitial fibrosis, increased the myofiber regeneration, and had a higher percentage of muscle satellite cell. This big work led to a publication in Science Translational Medicine, where we demonstrated that the TSHR blockage is responsible for the CNS cells, while the TSHR stimulation can revert this regenerating capacity and could be a therapy for Duchenne muscular dystrophy. Now, what we are doing, we are also see if together with the contribution of the satellite cell with the fibrogenic progenitor, with the FAPS, the adipogenic progenitor, we can also revert and ameliorate this satellite cell. And in order to study this, we performed analysis on the Duchenne muscular dystrophy in order to better characterize the behavior of the satellite cell. In these studies, what we did, we analyzed the muscle biopsy of the patient, a total of 24 muscle biopsy derived from patients with different mutation in the dystrophin gene or presenting an absence of dystrophin, as you can see here, and we performed different studies in order to quantify parameter of tissue adiposition and the presence of alteration of the PAX cell. What we have a dynamic in the patient with different picture at different stage. But what is clear is that already at the beginning of the disease, at 1-2 years, we have an alteration with important necrosis, with the presence of hypercontracted fibers, and the fibrosis is present already very early. You can see here the patient. But this fibrosis tends to be stable. It plateaued in our muscle biopsy from our patient. While what we found that these cells contributing to the adipose tissue deposition increased with the time at later stage. And while the regeneration less active was dropping by the age of 9 years. So what we can infer is that there is an acquisition of the satellite cell senescence phenotype also in muscle biopsy from patient with these two markers that are highly expressed. In conclusion, what we have that the regenerative capacity in the end muscle is diminished over time. The satellite cells are senescent and acquire progressively a senescent phenotype, and we have that the number of PAPs is proportional to fibrotic level. So, in order to study this, we got financed by the National Agency of Research to see if the fibroadipogenic progenitor can contribute with different population to this phenomenon. We can, if we can identify a pharmacological modulator of adipocyte ectopic deposition, combined to this forskolin treatment that could be applied to patients in order to treat the Duchenne muscular dystrophy. In conclusion, skeletal muscle is capable to regenerate through the satellite cell. In Duchenne, we have a loss of regenerative capacity, and although we found that there is an alteration with a senescence that is more important than the exact pathogenic mechanism should be clarified. The interaction with satellite cell and FABs could be a possible mechanism that we will explore with our research project, and the pharmacologic approach to restore muscular regeneration through satellite cell simulation are biopsy patient with Duchenne muscular dystrophy. These are the two persons that performed the majority of the experiment of this study, Valentina Taglietti and Nastasia Cardoni. I thank you very much, and I ready to respond to your questions.
Thank you very much, Professor Eduardo. Prof. Eduardo, hello?
Hello, yes, thank you.
Yes, thank you so much for this great lecture with updated knowledge. Thank you so much. Any questions for Prof. Eduardo?
Yes.
Yes, our Prof. Nagia Fahmi.
Professor Eduardo Malfacci, thank you so much for being with us. I know you are very busy, But it was very nice to see you with us, and next year we are waiting you physically. I have two questions. One
Yes.
about that thyroid stimulating hormone you used in therapy. Is there affection of the thyroid functions or other functions of the, I know it was an animal model until now. Is there any affection of other organs or the function of the thyroid hormones in the body? This is my first question.
No, we didn't find in the animal model any affection of the thyroid axis. And the forskolin is something that has been used before as a complement to boost and simulate the production of hormones. So it's something that is safe, and we are now performing more study with different dosage in cells in order to see if we can apply this complement. It wouldn't be a therapy that will resolve completely the phenotype of Duchenne. We now know that in Duchenne we will probably have a cocktail of therapies, but this could be an add-on that could ameliorate the phenotype.
Okay, another question about myoblast transfer, because I visited Professor Peter Lowe in the United States in cell therapy maybe several years ago, and he was working on this, and he worked on skeletal muscles and on cardiac muscle, and then his work stopped by Lowe, I think. I don't know if you know about this or not. It was about 1997, 98, and Peter Lowe has a cell institute about myoblast transfer, and even he has a book about this. What is new in myoblast transfer in the research of Duchenne?
So this approach that is viable from an experimental procedure is not, there is not much novelty, and the gene therapies approach, gene editing approach, has completely scotomize this research that is very interesting. Although I think there are several limiting factors. For example, the muscle mass is huge in our body, and you cannot transplant the equivalent of the deficient muscle mass in Duchenne muscular dystrophy on other dystrophy. Although I think it could be interesting for sparing or saving specific muscles that are very important for the function of the patient, and I mean, for example, the arm muscle to contact the wheelchair. So for me, there is still a place for this, but it's true that now in the pipeline we have many exon skipping agents, we have many gene therapies that are arriving. The read-through therapy has been stopped for now in Europe with Atalren, but the investment have been done on other procedure, although for me it remains a viable and interesting approach.
Okay, thanks. Thank you, Prophet Bardo. Another more question? Yes, Professor Hela.
Good afternoon. Many thanks for your lecture. I would like to ask about rationalization for sparing of extraocular muscles in your research. Is it related to muscle fiber type?
The little is known about the extraocular muscle in human. We are now starting to use the samples that are extracted during the strabism strabism surgery in order to have a baseline and comprehend if this TSH receptor is more expressed in type 1 fibers or type 2 fibers. When I examined the first samples that we received, I see that there is a type 2 fiber predominance, which is completely expected, as the extraocular muscle are the fastest muscle that we have, because they help us to focus on our fovea, the images, and to have a precise vision. And in the evolution of the human, this was necessary in order to spot possible danger and escape. So I think that there is a differential expression on the different muscle fiber type that could explain also their function and their different conformation in terms of satellite cell, but we need more knowledge and more experiments. So maybe next year we will have more data to show you.
Thank you so much, Prof. Another more question, if you please.
Yes. Hello,
Okay,
Dr.
introduce
Edward.
yourself, Professor. Introduce yourself.
My name is Dr. Ahmed Al-Husseini, lecturer of neurology, and I have maybe 3 questions for you, Dr. Eduardo. The first one, your work about fab cells, it is very great and very important. What about dystrophin expression?
Can you talk a little bit louder, because
Okay,
I get you.
First question, what about dystrophin expression in extraocular muscle? So, dystrophin extraocular muscle is normal or also affected in this is my first question. And what do you think about satellite cell and dystrophin? So we know dystrophin is absent in DMD patients. So in satellite cell dystrophin absence also, of course, satellite cell will be defect in dystrophin. This will affect the polarity and transplantation or not? And the third question, I want just, do you know, the last trial about the HOPE study, the stem cell transplantation for cardiomyopathy, now it is a new trial, so I want to know about your opinion about this study. And thank you very much.
Thank you. About the third question, yes, dystrophin is absent, as is expressed in all the skeletal muscle and also the cardiac muscle, and there is also dystrophin in smooth muscle, so it's absent. Concerning the association of the presence of satellite cell in extraocular muscle. So what we have seen that there is a sparing of the satellite cell that can regenerate, at least in the mouse model. We don't have, in the rat model, sorry, we don't have solid knowledge in the human that are able to regenerate and do not become senescent, and is in the limb muscle of the rat. Okay, this is due to this expression of hyperexpression of the TSHR, and now we are studying in the human sample in order to understand more. And this we will do through transcriptomic analysis and spatial transcriptomic that allow you to see in tissue the expression of the different RNA. Concerning your question about the cell transplant in the heart, I think this is very promising, because the heart is a less complex structure and a less big structure compared to the skeletal muscle mass. So we can target specific areas, for example, of the left ventricle, that can assist to ameliorate or prevent the happening of dilated cardiomyopathy. But still, we need to see if in terms of safety this is viable. So I'm very I'm looking forward to see the data and hopefully could apply this therapy to the patient. But still, I think it's a long way before we could inject this cell in the heart of our patient.
Okay, thank you so much, Professor Eduardo, for your knowledge and effort. See you next, inshallah, in Egypt.
Thank you, with pleasure. شكرا.
شكرا دكتورة ناجية على الدعوة الكريمة، وبصراحة مؤتمر النيورو ماسكولار من المؤتمرات القوية اللي كلنا بنبقى حريصين نحضرها، وبالتوفيق دايما، وبنختم السيشن عشان الصلاة.